Good evening, everyone. So today we are continuing with the lecture 3 for the unit cell, the unit of life. So in the last lectures, we discussed about what are prokaryotic cells, what are the structure of a prokaryotic, what is the structure of a prokaryotic cell. We give a brief introduction about a eukaryotic cell. So we study about how a plant cell is different from an animal cell. And now we are dwelling more into what is a eukaryote, what are the other components of a eukaryotic cell and what are their functions. So we know that in plant cells, there is an important structure which differentiates it from the animal cell, and that is the cell wall. So cell wall is a non-living rigid structure which surrounds the cell membrane, and it forms an outer covering for the plasma membrane of bacteria. As we see that in gram-positive bacteria, it is made up of peptidoglycan, the outer cell wall, and in case of gram-negative bacteria, we have lipopolysaccharide as a part of the cell wall. So cell wall is present as the outer covering of the plasma membrane of bacteria. In case of some protistans, in fungi also we know that the cell wall of chitin is present, and it is also present in algae and plant cells. So algae have a cell wall that is made up of cellulose, galactans, mannans, and minerals like calcium carbonates, while in other plants it consists of mainly cellulose, hemicellulose, pectins, and proteins. So for the plant cell wall, cellulose is the main constituent. Besides the other constituents, cellulose is the main constituent of the plant cell wall. And hemicellulose, cutin, pectin, lignin, sobrinin are also present in the cell wall of the plants. So the cell wall of the plant can further be divided into different types. Like it can be a primary cell wall, and then it can further lead to the formation of a secondary cell wall. So primary cell wall, as the name suggests, is a first cell wall, and it is present in the young plant cells. It is thin, elastic, and capable of growth. And the main composition of the primary cell wall is cellulose, hemicellulose, and pectin. But with the time, as the cell matures, primary cell wall gradually start diminishing and leading to the formation of the secondary cell wall around it. So secondary cell wall is formed on the inner side towards the membrane side of the cell that we will see in the diagram later. So it is present on the inner towards the membrane side of the cell. It is rigid and thick as compared to the primary cell wall. When on one hand we see that the primary cell wall is thin, elastic, secondary cell wall is rigid and thick. And the main composition of the secondary cell wall is cellulose, hemicellulose, pectin, cutin, lignin, and suberin. And secondary cell wall is absent in the meristem cells. So we know there are different types of cells which are present in the plants. So in case of meristem cells, secondary cell walls are absent. Now, both the cells, like two cells in a plant, are connected through a structure which is known as the middle lamella. And this middle lamella is basically also known as the glue that holds the different cells together. So it is also known as the plant cement. So the middle lamella is a layer mainly made up of calcium pectide. Its composition is really important because it is usually asked in the exams also, like what is the composition of middle lamella? So it is made up of calcium pectide, and which holds or glues, as I mentioned, that it acts as a glue, and so it holds the different neighboring cells together. It is a common layer between the two cells, and it is composed of calcium and magnesium pectides, thus acting as a plant cement. So although it has both calcium and magnesium, but the amount of calcium is more as compared to the magnesium. So if this is the representative of the primary cell wall and secondary cell wall, and how the different cell walls are arranged. So we can see that in the above diagram, mostly the primary cell wall has been shown. So, but with time, as the primary cell walls start diminishing, we see that there is a formation of the secondary cell wall, and the main composition of the secondary cell wall consists of hemicellulose, lignin we can see is the main constituent, and pectins. So we can see that the secondary cell wall is present just near to the adjacent to the plasma membrane, as we studied earlier. So this is how, and this is the blue color line represents the middle lamella, which helps in the binding or which helps in the connection of the, like that helps in as a cement or glue between the two neighboring cells. So this is how this primary cell wall or a secondary cell wall structure looks like. The next important structure in the cell wall is the plasmodesmata. So as I mentioned when I was describing about the animal cells and plant cells, so I mentioned one thing that plasmodesmata is a special structure which is present in the plant cells. Plasmodesmata is not present in the animal cells. And what is the function of plasmodesmata? It helps in the making the connections between the two cells, so that the transport of the materials can take place from one cell to the another cell. So the cell wall and middle lamella may be traversed by plasmodesmata. So these are just the connections. These are the cytoplasmic connections between the two neighboring cells. So we can see in this diagram that there is a cell wall which is present. Two cells are connected through middle lamella, which we mentioned earlier. And these two cells are connected through cytoplasmically through the connection which is known as the plasmodesmata. So cytoplasmic there can be two types of connections, which is symplastic and apoplastic. Cytoplasmic connections. So I'll repeat this again. So basically, we see that in this case, two cells are connected through a middle lamella. Now, the cytoplasmic connections which are present in between the two cells is known as the plasmodesmata. So plasmodesmata basically helps in the transport of material. They are the cytoplasmic connections that help in the connecting the two neighboring cells and allows the movement of materials from one cell to the another cell. Now, as we see in the diagram, this is the connection that allows the movement of the molecules from one cell to the another cell, and this is known as the plasmodesmata or plasmodesma. And it is present only in case of the plant cells and is not present in the and is absent in the animal cells. So the movement of materials can take place through two pathways. Either it can be an apoplastic pathway, or second, it can be a symplastic pathway. So in the figure, we can see that the orange line represents the movement of the material through the cell wall, and the bluish or purplish line represents the movement of the material through the cytoplasm. So whenever the movement takes place, this is very important concept because there are many questions which are asked from this. So what is apoplastic pathway and what is symplastic pathway is really important to understand. So it is very important that when the movement of the materials take place from the cell wall through the cell wall, then the pathway is called as the apoplastic pathway. And when the movement of the materials takes place through the cytoplasm, the pathway is known as the symplastic pathway. So there are certain differences between the apoplast and the symplast pathway. For example, the apoplast pathway comprises of, as I mentioned, the cell wall and the intercellular spaces. So the apoplastic pathway comprises of the cell wall and intercellular spaces, while symplast comprises of the cytoplasm or the protoplasm. The next difference is living parts versus non-living parts. As we can see that the apoplastic pathway deals mainly through the cell wall. So, and we know that cell wall is a non-living structure. So, we say that apoplast has non-living parts, whereas symplast has living parts, which is the cytoplasm. Also, pathway, we know that the apoplast makes a apoplastic pathway. Apoplastic pathway means from above the membrane, above the plasma membrane. So, that is through the cell wall. So, apoplast makes the apoplastic pathway, symplast makes through the cytoplasm, that is symplastic pathway. Now, rate of pathways. Apoplast pathways are faster than symplastic pathway. The reason for this is in the cytoplasm, we know that most of the cellular activities, as we mentioned earlier also, that most of the cellular activities takes place in the cytoplasm. And since most of the cellular activities are taking place in the cytoplasm, there are many metabolites that may be present in the cytoplasm that will hinder the movement of the molecules. So, due to this hindrance, the movement of the molecules may slow down, thereby decreasing the rate of the transportation. So, apoplastic pathway is therefore faster than the symplastic pathway. Next, metabolic state. It is kind of similar, which I mentioned just now, that metabolic state in the symplastic pathway interferes the movement of water, unlike in the case of apoplastic pathway. Next is transportation. In the plants with secondary growth, more water and iron are transported mainly through the apoplastic pathway in the cortex. Because in the secondary growth, we see that during the secondary growth, there is a formation of secondary wall also, and secondary cell wall also. And most of the water and ions are transported through that cell wall. And since the transportation is occurring through the cell wall, most of the transportation, we can say that it occurs through the apoplastic pathway in the cortex. Now, beyond cortex, water and ions are mainly transported through the symplastic pathway. So these are some of the basic differences. But most important thing to remember is that apoplastic pathway, name suggests apoplastic means above the plasma membrane, that is through the cell wall. So in apoplastic pathway, the transportation takes place through the cell wall, while in case of symplastic pathway, transportation occurs through the cytoplasm. So these are the basic differences in between both the apoplastic and the symplastic pathway. So this was the basic about the cell wall of the plant. So just for a quick revision, that the cell wall of a plant is made up of mainly cellulose, and they are of two types, primary and secondary. We see that with time, primary cell wall diminishes and secondary cell wall starts growing. And most of the secondary cell wall is adjacent to the plasma membrane, and both the two cells are glued or hold together. Two plant cells are glued or hold together by the connection, which is known as the middle lamella. Middle lamella is also known as the plant cement, or we can say that it is, and its main composition was the calcium pectide and magnesium pectide, and calcium was the main component. And then we see that the transportation of the water molecules or the ion molecules may takes place through a cytoplasmic connections, which are known as plasmodesmata in plants, and these transportation may occur either through symplast mechanism or the apoplast mechanism. And then we study what are the basic differences in between the symplast and apoplast. Now the next important component of a cell is a endomembrane system. So while we talk about different organelles which are present in the cell, there are some organelles that function in coordination with each other. So those organelles that function in coordination with each other and are mainly composed of similar kind of membrane, they are brought under the bigger umbrella term, which is the endomembrane system. And these, while each of the membranous organelles is distinct in terms of its structure and function, there are many of these are considered together as an endomembrane system because of the functions they perform with the help of each other. So the endomembrane system is made up of two words, like endo means within. So it is a group of membranes and organelles in eukaryotic cells that works together to change, package and transport the materials like lipids and proteins in and out. Endomembrane system includes endoplasmic reticulum, Golgi complex, lysosomes and vacuoles, because their functions are coordinated, that we will see how later. So these are the main parts of the endomembrane system. So the endomembrane system comprises of endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. And all these organelles function together with each other in coordination with each other. While there are many other organelles like mitochondria, chloroplast, peroxisomes that are also present within the cell. But since these do not function in coordination with each other or in coordination with the above mentioned organelles, therefore they are not considered as a part of endomembrane system. So it is very important that which organelles are considered as a part of endomembrane system, while which are the ones that are not a part of endomembrane system. So what are the organelles that are a part of endomembrane system? They are endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. So this is how the all the organelles which are a part of endomembrane system functions. So Basically, endoplasmic reticulum leads to the formation of the protein, or the smooth endoplasmic reticulum helps in the lipid synthesis. Then these part, these move to the complex like Golgi complex, or then they may be transported to the lysosome. So in coordination with each other, they help in the transportation of the cargo. They helps in the making of the cargo, they helps in the packaging of the cargo, and then they helps in the transportation of the cargo. So that is how they function in coordination with each other. So the first component of endomembrane system is endoplasmic reticulum. So what is endoplasmic reticulum? So the electron microscopic studies of eukaryotic cells reveal the presence of a network or reticulum of tiny tubular structures that are scattered into the cytoplasm, as we can see in this diagram also. So these are the structures that are scattered into the cytoplasm. They are mostly connected through the nuclear envelope, and they are scattered in the cytoplasm. And since they are made up of network-like structure or tiny reticulum-like structures, they are termed as endoplasmic reticulum. Endoplasmic reticulum divide the intracellular space into two distinct compartments: luminal, that is inside the ER, and extraluminal, as the name suggests, cytoplasmic compartments. So ER basically divides the overall intracellular space into two distinct compartments. The one which is the space which is present inside the ER, that is known as the luminal, and the one which is present outside the ER, which is the extraluminal, that is the cytoplasmic compartment. Now, what are the components of endoplasmic reticulum? Endoplasmic reticulum mainly comprises of three components. The first is cisternae, second is tubules, third is vesicles. All these three components vary in their shape and vary in their sizes. So cisternae, as we can see in this diagram also, they are long, flattened and unbranched units that are arranged in the stack-like manner, like one above the other. So cisternae are long, flattened and unbranched units arranged in stacks. Tubules are usually irregular. They are tube-like structures, and they are often branched tubes bounded by the membrane. So we can see here, this is the diagram of the tubules. They are usually branched tube-like structures, and they are surrounded by the membrane. Vesicles, as we usually know about what are vesicles. So vesicles are oval membrane-bound structures. So there is a difference in between the morphology, how these three different components look like. So cisternae are long, flattened and unbranched units, while tubules are cylinder, branched tubes-like structure, and vesicles are oval membrane-bound structures. So this is the basic structure of endoplasmic reticulum. We can see that it is composed of cisternae here. These are the tubules we can see. So, and vesicles are formed when something is pinched off. Like in the last class also we used the term vesicle, and in the figure we see that when something is pinched off through the plasma membrane, it forms the vesicle. So maybe later we will see vesicle somewhere in the diagram. So in this diagram, we can clearly see these are the cisternae and these are the tubules. So this is the diagram of endoplasmic reticulum, and in this diagram, we can clearly see that there are two types of endoplasmic reticulum. One which is the rough endoplasmic reticulum and one which is the smooth endoplasmic reticulum. In the diagram itself, it is very clearly represented that in case of rough endoplasmic reticulum, a structure which we also discussed in the last class, ribosome. So a structure which is known as the ribosome is present, giving it a rough appearance, thereby giving it the name rough endoplasmic reticulum. While the structures on which ribosome is not present, they are smooth and thereby known as the smooth endoplasmic reticulum. So endoplasmic reticulum is of two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. This is on the basis of what? Whether the ribosome is present on the surface or ribosome is not present on the surface of the endoplasmic reticulum. If it is present, it will be known as the rough endoplasmic reticulum or RER. And if it is absent on the surface of the endoplasmic reticulum, it is known as the smooth endoplasmic reticulum or SER. one important thing to notice here is that I would like to mention is the ribosome which is present in the eukaryotes. So we studied that the ribosome which was present in the prokaryotes was 70S. That is, its Swedberg unit is 70S. While in case of eukaryotes, it is not 70S, it is 80S ribosome which is present. And the main function of ribosome, as we see in the earlier lectures also, it helps in the protein synthesis. It attaches to the mRNA and helps in the protein synthesis. So we will see that the presence of the ribosome on the surface of the rough endoplasmic reticulum thereby imparts the function of protein synthesis to RER. So studying in detail about rough endoplasmic reticulum structure, rough endoplasmic reticulum is named so because of its appearance, as I mentioned, and it has flattened sacs having a few ribosomes on its external surface, and subsequently the name. So we can see that mostly the ribosomes are present on the flattened sac-like structure, while in tubules-like structure, ribosome is absent. So most of the composition of the RER is what type of structure? It is mostly cisternae, as we discussed just now. What is the difference between cisternae and tubules? So we can clearly see that mostly the ribosomes are present on the flattened sacs-like structure, which are known as cisternae. So rough endoplasmic reticulum is mainly composed of cisternae, and it is usually associated with the synthesis and secretion of proteins in the liver, chemicals, and different substances in the glands. Rough endoplasmic reticulum is are the place where the protein synthesis occurs. Now, what are the functions? So, the elements of rough ER is related with the protein synthesis. Rough endoplasmic reticulum likewise assumes a crucial part in protein folding. So, since the protein synthesis is taking place there, one of the most important function of RER is that it plays an important role in protein folding. Like, if protein folding does not occur properly, then there are certain diseases, for example, amyloid, or we know about Alzheimer's. So there are certain diseases that may take place if protein folding does not occur properly. So rough endoplasmic reticulum is also known to play important role in protein folding. Therefore, it additionally makes the important quality control system. The second most significant capability after protein synthesis and protein folding is protein arranging. And so therefore, the functions of RER is not only protein synthesis, not only quality control of the protein, but also helps in the arranging of protein. And it occurs abundantly in the cells which are actively engaged in protein synthesis and secretion. The next is smooth endoplasmic reticulum. So we discussed about what is rough endoplasmic reticulum. Now, what is smooth endoplasmic reticulum? Again, something which does not have ribosomes. As I mentioned, smooth endoplasmic reticulum has a tubular structure mainly, and it has a different function. Since ribosomes are not present, it is not taking part in protein synthesis. It is taking part in the development of phospholipids, the main lipids in the cell membranes, and are essential during the digestion. Smooth ER transports the results of the rough ER to the other cell organelles, particularly the Golgi device. As I mentioned that ER works in coordination with Golgi, which further works in coordination with lysosome. So basically rough ER leading to the formation of proteins. Smooth ER transports those proteins to the other organelles, especially to the Golgi body where the packaging takes place. So this is how this endomembrane system works in coordination with each other. As we see, rough ER is mostly present in the cells where the protein synthesis is important. Smooth ER is present in the cells, is abundant in the cells concerned with the glycogen and lipid metabolism, since they are associated with the lipid metabolism and lipid synthesis. Now, what are the functions of SER? SER is essential in the synthesis of essential lipids such as phospholipids and cholesterol. Smooth ER is also responsible for the secretion of steroid hormones. It is responsible for the metabolism of carbohydrates. The smooth ER stores and release calcium ions, and therefore, since we know that calcium ions are very important from nervous system point of view and from muscular system as well. So these are important for the nervous system and muscular systems. Now, what are the overall function of endoplasmic reticulum? So rough ER is frequently observed in the cells actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. So this is what I was mentioning also, that these are connected to the nuclear envelope, and these are the extensions that occurs or protrudes from the nuclear envelope. So this is what I mean to say here when I say they are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is a major site for the synthesis of lipids, and in animal cells, lipid-like steroid hormones are also synthesized in SER. Besides this, they also act as a cytoskeleton. They provide the support, mechanical support to the cells. So microfilaments, microtubules, and ER forms the endoskeleton of the cell. Since they are network-like structures, they provide the support to the cell, therefore providing the mechanical support, and forms the endoskeleton of the cell along with microfilaments and microtubules. It helps in the intracellular exchange. So ER forms intracellular conducting system, as we discussed just now also. Transport of materials in cytoplasm from one place to another may occur through the endoplasmic reticulum. And at some places, ER is also connected to plasma membrane. So ER can secrete the materials outside the cell. So therefore, ER also plays an important role in transportation of materials or transportation of cargo in and outside the cell. The second important component of the endomembrane system besides ER is Golgi apparatus. Now, Golgi apparatus is named behind the scientist who discovered it. So, Camillo Golgi, who first basically observed it. So, Camillo Golgi in 1898 first observed the densely stained reticular structure near the nucleus, and these were later named Golgi bodies after him. They consist of many flat disc-shaped sacs, or which are also known as cisternae, and which are around 0.5 to 1 micron in diameter. These stacks are parallel to each other and very different, like in different cells, in different Golgi complex, varied number of cisternae may be present. These Golgi cisternae are concentrically arranged near the nucleus. So there are two faces of these cisternae. We can see that these are the cisternae. These are arranged parallel to each other. And there are two faces. One is the cis phase and one is the trans phase. So what is cis phase and what is trans phase? So the convex surface of the cisternae which is towards the nucleus. So the one which is convex and which is towards the nucleus is known as the cis phase, while the concave surface of the cisternae which is towards the plasma membrane is known as the trans phase. Trans phase is also known as the maturing phase, as most of the matured items or the mature cargo is released through the trans phase. So it is the maturation phase. It is towards the plasma membrane so that more cargo, cargo can be released out of the cell if needed. So cis phase is towards the nucleus. It is convex in shape. It is present towards the nucleus. And the trans phase is present towards the plasma membrane. It is concave in shape. And the other thing is that the trans phase is also known as the maturing phase. The cis and trans phases of the organelles are entirely different, but they are interconnected. So the Golgi apparatus primarily performs the function of packaging materials to be delivered, as I mentioned, either to the intracellular targets or outside the cell. So basically the main function of the Golgi apparatus is packaging. So whatever the proteins and lipids are formed by the ER, they are transported to the Golgi apparatus for the further packaging and transportation. And so that after packaging, the material or the cargo can be delivered either intracellularly or outside of the cell, whatever the requirement is. And materials to be packaged in the form of vesicles from the ER fuse with the cis phase of the Golgi apparatus and moves towards the maturing phase. This explains why the Golgi apparatus remains in close association with the endoplasmic reticulum. So as we see that Golgi apparatus is present very close to the ER reticulum. So what happens is whatever the proteins, like whatever the cargo is formed by the, like whatever is formed by the ER, it is delivered in the form of vesicles to the Golgi complex, where these vesicles fuse with the cis phase of the Golgi complex. Then they undergo maturation and packaging and are delivered outside of the Golgi complex through the trans phase. A number of proteins synthesized by ribosomes on the ER are modified in the cisternae before they are released from its trans phase. So sometimes protein modification may also takes place in the Golgi complex depending on the type of protein. Golgi apparatus is the important site for the formation of glycoproteins and glycolipids. So this is basically, you can say it is a kind of post office where the Golgi apparatus modifies, sorts, packages proteins for the secretion. Like which protein is to be transported where. Just like what a post office do, it sorts our materials, like which of the material is to be delivered where. Same happens inside the cell. So this is done by the Golgi complex, which is also, you can say it's kind of cell's post office, where it modifies, sorts and packages proteins for the secretion and decide the fate of each protein. The next component of endomembrane system is lysosomes. These lysosomes are the membrane-bound vesicular structures which are formed by the process of packaging in the Golgi apparatus. So we can see in this diagram that this is just a representative that in a membrane-bound structures, there are many enzymes, mainly acid hydrolases, which are present. For example, lipases, which breaks down the lipids, proteases that break down the protein, carbohydrates that breaks down the carbohydrates, and nucleases that breaks down the nucleic acid, that is DNA and RNA. So the isolated lysosomal vesicle have been found to be very rich in almost all types of hydrolytic enzymes. As I mentioned, hydrolases, lipases, proteases, and carbohydrates that becomes active at the acidic pH. So the pH of the lysosome is different from the outside environment. That is, its pH is quite acidic, around 5. And the membrane of the lysosome has active proton pumps. And these proton pumps, active proton pumps means they require energy. And by the help of utilizing this energy with the help of this protein pump, there is the influx of the protons or the H plus ions into the lysosome. And since there is an influx of H plus ions, more the H plus ions, more acidic the environment is. So therefore, this helps in maintaining the acidic pH in the lumen of the lysosome. Lysosomes are highly polymorphic cell organelles, like they have different morphologies, and because lysosomes have different physiological states. For example, lysosome may be present itself as a lysosome, or it can be present as a, in combination with phagosome, it may lead to the formation of a phagolysosome that has a different pH. So based on the different physiological states, lysosomes are basically polymorphic, and they acquire different pH under the different physiological state condition. So there are different types of lysosomes. They can be classified mainly of 3 types. Primary lysosomes, which are also known as storage granules. The second is these secondary lysosomes. Third is tertiary lysosomes or residual bodies. So primary lysosomes or storage granules, as the name suggests, these lysosomes stores enzymes. For example, the one which I show here. So these are the primary lysosomes that store the enzymes, acid hydrolases in the inactive form. And these are newly formed lysosomes. So primary, as the name suggests, these are just the new ones, and they are the newly formed lysosomes. and they store the enzyme acid hydrolases in the inactive form. Secondary lysosomes are further can be of two types, heterophagosomes, as I mentioned, and autophagosomes. So heterophagosomes are also known as digestive vacuoles and digestive lysosomes. Autophagosomes are known as autophagic lysosomes. So in case of digestive vacuoles or heterophagosomes, these lysosomes are formed by the fusion of primary lysosomes and phagosomes. The one which I mentioned, like under different physiological states, the lysosome may acquire different morphologies. So the one is heterophagosome. So heterophagosome, how the heterophagosome formation takes place? When the lysosome formed by the fusion of primary lysosome and the phagosome, these are called as the secondary lysosomes. And the second one is autophagic lysosomes or autophagosomes. So lysosomes containing cell organelles to be digested. Autophagosome means cell. So when the lysosomes that contains the cell organelles to be digested, like cell organelles to be digested, it forms the autophagosome. Like for example, to make it easy and make it more clear, if the lysosome helps in the digestion of its cell organelles, it leads to the formation of autophagosome. While if it fuses with the phagosome and helps in the digestion of the food molecules that is taken up by the cell by the process of endocytosis, it leads to the formation of heterophagosomes. The third is tertiary lysosomes or residual bodies. Lysosomes containing undigested material. As the name suggests, residual bodies means the remaining ones. So the lysosomes containing undigested materials are called residual bodies. And since these are undigested material, they are thrown out of the cell. They are excreted out of the cell by the process known as exocytosis. So these are the three basic different types of lysosomes. The one is the primary lysosomes. The second one is secondary lysosome. Third one is residual bodies or tertiary lysosomes. So lysosomes are also known as suicidal bags. Why? Because they eat up their own cell organelles, thus leading to the cell death in which they are itself residing. So they are also known as suicidal bag. So as I mentioned, in case of secondary lysosome, heterophagosome may form or autophagosome may form, thus leading to the process of heterophagy and autophagy. Heterophagy means eating others, while autophagy means self-eating. So in case of heterophagy, we can see that suppose this is a food particle which is shown here in the brown color that comes and is taken up by the cell. So basically it is taken up by the process of endocytosis inside the cell, leading to the formation of a food vacuole inside the cell. Now what happens is, to digest this food we need hydrolytic enzymes. So lysosomes that contain the active hydrolytic enzyme or the digestive enzymes comes and fuses with this food vacuole to act. So this leads to the formation of this step. At this step where the lysosome fuses with the food vacuole, this structure is known as the heterophagosome that we just discussed. So this heterophagosome, with the help of these enzymes, these hydrolytic enzymes digest the food particles, thereby helping in the digestion of the food. So this is how, what is heterophagy and what do I mean by heterophagosomes. Now, what is autophagy and what is autophagosomes? So basically within the cell, so there are certain organelles which are present inside the cell. So lysosome fuses with the vesicles containing these damaged organelles. So these enzymes which are present in the cells, these hydrolytic enzymes digest the organelles, different organelles which are damaged, and since it fuses with the vesicle that has its own organelles, it is known as the autophagosome. So basically, in this case, autophagosome is formed and autophagy leads to the digestion of cell organelles. So this is the basic difference between heterophagy and autophagy. So this was about the lysosome. The last component of the endomembrane system is the vacuole. So till now, what we study in the endomembrane system, we study what is endomembrane system, what are different components like endoplasmic reticulum, lysosome, Golgi complex, and the last one is vacuole. We study what is endoplasmic reticulum, what are the functions, what are the different types of endoplasmic reticulum, how it functions in coordination with the Golgi complex, and how then these function together in coordination with the lysosome. So next is the vacuoles. So vacuoles are the membrane bound space found in the cytoplasm. It contains water, sap, excretory product and other materials that are not useful for the cell. In case of plant cells, the vacuoles can occupy up to the 90% of the volume of the cell, shifting the nucleus towards the periphery. While in case of animal cells, usually the size of vacuole is smaller. The vacuole is bound by a single membrane which is known as the tonoplast. So in this we can see that in this diagram, this is the vacuole which is surrounded by a blue colored line membrane which is known as the tonoplast. So tonoplast basically helps in maintaining the osmolarity of the, like if there is an imbalance in the osmolarity outside of the vacuole and inside of the vacuole, then the bursting of the vacuole may takes place. So tonoplast basically helps in maintaining, facilitates the transport of ions and materials against the concentration gradient into the vacuole, and hence their concentration is significantly higher in the vacuole as compared to the cytoplasm. So we can see that here the energy is utilized since the transportation is taking place against the concentration gradient. So it is an active transportation mechanism. And we can see that since it is transporting against the concentration gradient, therefore the concentration of the materials and the ions is comparatively higher in vacuoles as compared to the cytoplasm. In organisms like amoeba, the contractile vacuole is formed, and it is very important for the process of secretion and osmoregulation. In many cells, for example in protists, food vacuoles are formed by engulfing the food particles. So there are different types of vacuoles which are present in different organisms. So we see that in case of plants, the vacuoles occupies mostly the major space of the plant cell, may occupy up to 90% of this volume of the cell, while in case of animals, vacuole is comparatively smaller. In amoeba, vacuole has a very important function. It has a contractile vacuole that helps in the excretion and osmoregulation. In cells like protists, food vacuoles are formed by engulfing the food particles. So this is what the vacuole is. So based on the discussion we had today about the endomembrane system, there are certain questions that I would like to discuss here. The first is primary cell wall is option 1, thin, flexible, capable of growth, and option 4 is all the above. So we study that the primary cell wall is quite thin and flexible and it has the ability to grow. Therefore, all the options regarding the primary cell wall are correct. Like it is thin, it is flexible, it has the capability to grow. Therefore, all of the above is the right option. Question number 2 is common layer between two adjacent plant cells. So we study study what is the common layer between the two adjacent plant cells is known. What is the glue? What is the substance that holds together the two adjacent plant cells? So the options are primary cell wall, option 2 secondary cell wall, option 3 middle lamella, and option 4 is the plasma membrane. So we know that the middle lamella is the structure that glues or that holds together the adjacent plant cells. So therefore, the correct answer is middle lamella. So middle lamella, which is mostly composed of calcium and magnesium pectate, majorly of calcium, is the component that is a common layer between the two adjacent plant cells. The next question is, which of the following is incorrect for cell wall? So the question is asking which is incorrect. So the first question is protect cell wall protects cell from mechanical damage. It helps in cell-to-cell interaction. It provides barrier to undesirable macromolecules, and it does not give shape to the cell. Since we know that cell wall is the outermost layer of the plant cell, and we know that plant cell wall actually protects them from the mechanical damage, as it is quite hard, and it helps in cell-to-cell interaction, as we know that middle lamella helps in attaching the two cells. Also, there are certain connections like plasmodesmata from the cell wall that helps in the cell-to-cell interaction. Also, it provides a barrier to the undesirable molecules, macromolecules. But the incorrect point is that it does not give shape to the cell, as one of the main functions of the cell wall is that due to its rigid nature, it provides a definite shape to the cell. So therefore, the correct, which one of the following options is incorrect for the cell wall? So the statement stating that it does not give shape to the cell is incorrect. Next question is, endoreticulum divide the intracellular space into two distinct compartments. So we know that as we studied that endoplasmic reticulum divides the intracellular space into two distinct compartments. One which is the space within the endoplasmic reticulum, and the second is the space outside the endoplasmic reticulum. So the one which was within the endoplasmic reticulum was known as the luminal space, while the one which was present outside the endoplasmic reticulum was extraluminal space, that is outside the lumen. So it divides the intracellular space into two distinct compartments, that is luminal and extraluminal. So the correct answer is option number 2, luminal. Next question is, components of endoplasmic reticulum are. Now, what are the different components of endoplasmic reticulum? So option number 1 is cisternae, option number 2 is tubules, option number 3 is vesicles, and option number 4 is all of the above. So we know that all the three components like cisternae, tubules, and vesicles are present in the endoplasmic reticulum. Cisternae, water cisternae, I am repeating it again for a revision, quick revision. So cisternae are sac-like structures that stack above each other. Tubules are branched cylinder tube-like structures, while vesicles are oval membrane-bound structures. So all of these three components are present in the endoplasmic reticulum. Next is RER, rough endoplasmic reticulum, has what kind of ribosome attached to it? So I mentioned that in case of rough endoplasmic reticulum, in case of eukaryotes, it is the 80S ribosome which is attached to the rough endoplasmic reticulum. So RER has 80S ribosome attached to it. The next question is, SER is involved in. So we know that smooth endoplasmic reticulum does not have ribosome, and it is not, thereby not at least involved in protein synthesis. So what are the other options? The first is protein synthesis, second is lipid metabolism, third is glycogen metabolism, and fourth is both 2 and 3. So we know that smooth endoplasmic reticulum is involved in both lipid and glycogen metabolism, and it is not at all involved in protein synthesis. So the correct option is option number 4, both 2 and 3. That is, smooth endoplasmic reticulum is involved in both lipid and glycogen metabolism. Next question is, Golgi complex was discovered by. So it is quite an easy question. Why? Because we know that it is named after the scientist who first observed it. So the options are, first Robert Brown, second Sheldon, third Camillo Golgi, and fourth is Schwann. So we know that it is the correct answer is Camillo Golgi, who first discovered or first observed the Golgi complex, and so the correct answer is option number 3. Next question is, which of the following is correct about cis phase of Golgi complex? So what is a cis phase? Which phase of the Golgi complex is cis phase? The one which is present near to the nucleus side and or the endoplasmic reticulum side, and cis phase is convex in shape. while the other phase was known as the trans phase, which was more towards the plasma membrane, and it is a concave shape, and it was also known as the maturation phase. So what are the options which are correct for the cis phase of the Golgi complex? The first is it is a forming phase. Second is it is a maturing phase. It is distal to the nucleus. And fourth is vesicles are released from it. So we know that correct answer is it is a forming phase. Forming phase matlab it is one from where the endoplasmic reticulum, the vesicles which are released by the endoplasmic reticulum enters the Golgi complex. So it is a forming phase, while the other options like it is a maturing phase. So no, it is not a correct answer. Why? Because the trans phase is the maturing phase of the Golgi complex. Similarly, it is not distal to the nucleus. As I mentioned, cis phase is the one which is present in proximity, that is in close to the nucleus, that is present closely to the nucleus, while distal to the nucleus, trans phase is the one which is present in distance to the nucleus. So also vesicles are not released. Vesicles are actually transported inside the Golgi complex through the cis phase. It is the trans phase through which the vesicles are released. So the correct answer about the cis phase of the Golgi complex is that it is a forming phase. Sorry. The next question is, Golgi complex is important site of formation for the both of, the correct answer is, the options are glycoproteins. Second option is glycolipids. Third option is polysomes. And fourth option is both 1 and 2. So we know that Golgi complex is an important site for the formation of glycoproteins and glycolipids. All the proteins and lipids which are synthesized by the ER is getting transported into the Golgi complex, where certain glycogen molecules, or you can say certain carbohydrate molecules, so sugar moieties are attached to the proteins and the lipids, leading to the formation of leading to the modification of the protein and formation of glycoproteins and glycolipids. So the correct answer is both 1 and 2. While polysome is a structure that is formed by the presence of many ribosomes on a mRNA, which we studied in the last class. So polysome is not the right answer. Golgi complex is the important site for the formation of glycoproteins and glycolipids. The next question is, membrane-bound vesicular structures which are formed by the process of packaging in the Golgi apparatus are lysosomes, RER, SER, or ribosomes. So what are those membrane-bound vesicular structures? We study that lysosomes are the membrane-bound vesicular structures that are sometimes formed by the process of packaging, during the process of packaging in the Golgi apparatus. These lysosomes contain hydrolytic enzymes and that are usually active at a lower pH and thereby help in the digestion of various components within the cell. The next question is, acid hydrolases found in the lysosome function at. So name itself suggests that these are the hydrolases. These are the lytic enzymes that act at an acidic pH. So from the options, we can clearly see that the option number 3, which is pH 5, is acidic. So acidic hydrolases found in the lysosomes function at an acidic pH of around pH 5. So the correct answer is option number 3, that is pH 5. The next question is, which of the following contains water, sap, excretory product and other materials which are not useful for the cell? So one by one, if we will see the functions of the options, following options. So rough ER is basically involved in the protein synthesis. Lysosomes mainly contains the hydrolytic enzymes that helps in the digestion. SER is something which is smooth endoplasmic reticulum, which is mainly associated with the lipid synthesis, while the vacuole is the structure that contains the water, sap, other excretory products or the materials which are not useful for the cell. So the correct answer of this option of this question is option number 4, vacuole. The next question is, membrane of the vacuole is called as. So we know that chloroplast are the structures that are the plastids that help in the during the process of photosynthesis. So tonoplast is the membrane of the vacuole, while cell wall is the the structure that is present around the cell membrane and it surrounds the complete cell, while cell membrane is the structure that provides a boundary to the cell. So tonoplast is the membrane of the vacuole. So the correct answer is option number 2, tonoplast. With this, I would like to end today's session, and if there are any doubts or any questions, I would be happy to answer those questions. And if there is any doubt in any of the questions that we discussed, so we can discuss that also. If there are no doubts, uh, we will take up the other topics, other organelles in the next class. And if there are no doubts, I would like to end the session.